Hello everyone, welcome to this session. In this session we have to discuss Unit 1, Diversity in Organisms. Chapter 2, Part 1 already we have discussed. Today we are going to deal Part 2 of this, and in the end of this chapter definitely we have to discuss the questions given in NCERT exemplar and the previous year examination questions. So this Part 2 that will deal, that is structure of monoarians, especially bacteria, the shape of bacteria, and then different type of nutritions and different type of, that is, respirations found, on the basis of which we will say that bacteria is, these are very simple in structure and very complicated in behavior. They have metabolic diversity. Yes, this is Part 2 of chapter number 2, that is biological classification. In Part 1 we have discussed, that is 5 kingdom classification, its merits and demerits. I told you that time also, that is 6 kingdom classification have been proposed now. This 6 kingdom classification that is proposed by Carl Woese. He divided kingdom Monera into 2 kingdoms, that is kingdom 𝕍𝕀𝔻𝕼 and kingdom 𝕍𝕀𝔻𝔼. Because both of these are monoarians, they are prokaryotics. But you see, 𝕍𝕀𝔻𝔼 differs from 2 bacteria, that is, the cell wall and the plasma membrane are different. That is on the basis of some major differences, such that is absence of peptidoglycan in cell wall of 𝕍𝕀𝔻𝔼 and occurrence of branch chain lipid in members of 𝕍𝕀𝔻𝔼. Because of these 2 important characters, 𝕍𝕀𝔻𝔼 have been separated from, that is, 𝕍𝕀𝔻𝔼. And thus we have, that is 6 kingdoms now, that is kingdom 𝕍𝕀𝔻𝔼, kingdom that is bacteria, is it? Then we have, that is kingdom that is protista, then kingdom fungi, kingdom 𝕆𝕋 and kingdom 𝕆𝕋𝔼. One more important thing, that he proposed 3 domains of life. According to him, that is the 3 domains, all the organisms have been kept in 3 domains. The one domain is archaea, one domain is, that is, bacteria, one domain is, that is, eukarya. Domain archaea, it includes the monoarians which do not have peptidoglycan wall and in which the cell membrane is having this branch chain lipid compounds. That is the kingdom 𝕍𝕀𝔻𝔼. In domain bacteria, that is 𝕍𝕀𝔻𝔼 kingdom have been kept. In case of, that is, domain eukarya, there are the remaining 4 kingdoms, that is the protista, then this is kingdom fungi, then this is kingdom that is 𝕆𝕋, and then this one is, that is, kingdom 𝕆𝕋𝔼. The 3 domains have been classified or separated on the basis of 16S ribosomal RNA. You see, ribosome is having the smaller unit, 70S ribosome. In a smaller unit we have this 16S ribosomal, that is RNA. The gene sequence are different, and all 6 kingdoms, they have been grouped on the basis of this into 3 domain: bacteria, archaea and eukarya. He also told this thing, that is, all organisms that have been originated from a common ancestor, that is progeny, whose idea of evolutionary relationship this is. And from this progeny, that is all these, that is 𝕍𝕀𝔻𝔼, that is eukaryotes and 𝕍𝕀𝔻𝔼, they have been evolved. Is it? Now we have to deal in detail, that is one by one. That one is, that is kingdom Monera. You see, kingdom Monera, protoplasm is not differentiated into cytoplasm and true nucleus. In case of monoarians, you know, that is nucleus, true nucleus is absent. There is no nucleus membrane. There is no distinction in cytoplasm and the nucleus. The sap vacuoles are absent, which are present in eukaryotes. We do not have here, and that's why the cyclosis is absent here. You see, in case of eukaryotes, there is 2 nucleus and the vacuoles are there. Cytoplasm to circulate, that is around these vacuoles, which is known as cyclosis. In case of prokaryote, this is absent. Presence of membrane-bounded organelles, these are absent. That membrane-bounded organelle, that this is mitochondria, that this one is chloroplast, that this one is Golgi bodies, that this one is endoplasmic reticulum, and so on. These are membrane-bounded organelles, these are absent. Only what is present here, that is 70S ribosome is present, and none of the other cell organelles is here. That is presence of primitive nucleus called nucleoid or genophore or incipient nucleus. It is composed of naked DNA in which that is histone is absent. You know, histone is absent in case of that is DNA of prokaryotes. Now, the shape of bacteria, that is common recognized 4 types of bacteria. These are spherical, known as coccus, plural will be cocci. Bacillus or bacilli will be the plural, rod-shaped one. Rod-shaped bacteria, this is the most common type. That is vibro, comma-shaped these are, and spirillum, these are spiral-shaped. These are the 4, that is the specific shape of the bacteria. Cocci spherical, these are rod-shaped, that is bacillus. This is spiral, this one is comma-shaped. This question once came, which is the most common shape of bacteria? That is bacillus one. Right? Now, come to the next. Bacterial life processes. Children, you see, this question is very important. It has appeared many times. And in NCERT, this is being highlighted. You see, the statement is, bacterial structure is very simple. This is prokaryotic one. It is very simple one. But their behavior is extremely complex. As compared to many other organisms, bacteria shows most extensive metabolic diversity. I repeat, behavior-wise, that is, bacteria is structure-wise, it is very simple, but its behavior, metabolic diversity is there. It is very complicated one. How to justify it? On the basis of 2 things. One is that is respiration, right? Respiration, and then we have to see that is mode of nutrition. We have to justify this statement that the bacteria is simple in structure and complicated in its behavior, right? You see, bacteria has all type of respiration, aerobic and anaerobic respiration. You are familiar with the term aerobic respiration. It needs oxygen. Anaerobic respiration, it doesn't need oxygen for respiration. But the subcategories of these are, one is obligate aerobes. They can survive only in presence of oxygen. Where are the obligate aerobes? Without oxygen, we cannot survive. So bacteria, some of the bacteria are obligate aerobes, cannot survive only in presence of oxygen. The example is Bacillus, that is subtilis. Then the second one is facultative anaerobes. What do we mean by? These bacteria, normally these are aerobic, usually aerobic these are. But if the oxygen is absent, then these are adapted to respire even in absence of oxygen. They can survive an aerobic condition also if sufficient oxygen is not present. Is it clear? This is aerobe, this is facultative anaerobes are, usually these are aerobic, but they can survive without oxygen also, that is in an aerobic condition. The example is Pseudomonas. The third category is obligate anaerobes. They don't have any option. They can survive only, that is in absence of oxygen. These are strict anaerobe, can survive anaerobically only. Oxygen is absent only, then they can survive. Clostridium, that is botulinum. In presence of air, oxygen, they die. The fourth one is facultative aerobes. You see that you compare this and this. Facultative aerobes, opposite to this, that is normally these respire, that is aerobically, but they can survive in presence of oxygen. You see, that is mistake is there. What we mean by, say, facultative aerobes means, how to define it? What are facultative anaerobes? These are, that is aerobes. Facultative aerobes means that these are usually aerobic. These are aerobic, but they can survive even, that is also survive in presence of oxygen. This one is, that is 𝕆𝕆𝕆. Is it very much clear? Should I repeat? Facultative and aerobe, these are, yes, usually aerobic. Facultative aerobes, usually these are anaerobic. I have to correct it here, that usually these are anaerobe, but means they do not need oxygen, but even if oxygen is there, they can survive in this. It's a very important chart. This one is, that is this one. Come to the next now. Nutrition in bacteria. Some of the bacteria are autotrophic, but majority of the bacteria are heterotrophs. Important question. Heterotrophs means bacteria are unable to make their food. So most of the bacteria are heterotrophs. Only a few are autotrophic one. You have to pay attention to this question. Now, we have to deal separately. First, we have to see autotrophic nutrition. Autotrophic nutrition. Autotrophic bacteria means what? That is, they use solar energy. First, we have to see here, that is autotrophic nutrition. Yes, these are photoautotrophic bacteria. Autotrophic, they will have two categories. One is which utilize solar energy. These are autotrophs. And the second one which do not use, that is light energy, but they can prepare their food. They will be called as chemoautotrophs. So this one is photoautotrophic bacteria. They use solar energy, and to trap this solar energy, they have the pigment. The pigment is bacteriochlorophyll, also known as bacteriopurpurin, and that is bacterioviridin. Bacterioviridin is also known as, that is chlorobium, that is chlorophyll. Chlorobium, that one is chlorophyll. So you see, we are dealing now, that is detail of photoautotrophic bacteria. See the chart here. Nutrition in bacteria. Why metabolically diversity is there? All type of nutrition present on Earth's planet are present in bacteria. You see here, that is, this is photo, we are dealing now, that is, these are that is autotrophs. Photoautotrophs, which use, that is light energy, solar energy is being used. There will be of two types. One is photolithotrophs, another will be photoorganotrophs. You see, children, that in photosynthesis, the source of electron and hydrogen ions, proton, is very important. Photolithotroph, the source of hydrogen and electron is inorganic sulfur. This is inorganic sulfur compounds these are. While in case of photoorganotrophs, yes, the source of electron and hydrogen, this is non-sulfur organic compound. Is it? Both they will use, that is solar energy. Now see the part of this. Photolithotrophs, that is two types. One is purple sulfur bacteria, one is green sulfur bacteria. The pigment present here is bacteriopurpurin. Here the pigment is, that is bacterioviridin, also known as, that is chlorobium, chlorophyll, as I told you. What is the difference? That they use inorganic sulfur compounds, that is hydrogen sulfide is the source for electrons and protons. The example is chromatium and thiospirillum. Green sulfur bacteria, that the source is, that is they use what? Yes, other than, see, that other than hydrogen sulfide, that is source is other than hydrogen sulfide, other sulfur compounds, that is, these are used as source of electrons and that is protons. Here, they use hydrogen sulfide as source of, that is, hydrogen and that is electron. Example is 𝕆𝕆𝕋𝔼𝕆. Is it very much clear? They use inorganic sulfur compounds that will use any other except hydrogen sulfide. It will use hydrogen sulfide. Photoorganotropes. These are also of two types. Purple non-sulfur. You see purple sulfur these are. This is purple non-sulfur and this is green non-sulfur. They do not use sulfur compounds. The pigment here is bacteriopurpurin and here the pigment is bacterioviridin. Example is 𝕆𝕆𝕍𝕀𝔻𝔼 and this is 𝕆𝕆𝕍𝕀𝔻𝔼 and this is 𝕆𝕆𝕋𝕍𝕆 and 𝕆𝕆𝕋𝔼. This is about photoautotropes. You have to remember example, chromatium, thiosperillum and chlorobium. This is one group, photolithotropes, rhodospirillum, rhodopseudomonas, 𝕆𝕆𝕋𝕍𝕆 and 𝕆𝕆𝕋. This is another group, but all these, they belong to the same group, that is, they can do photosynthesis, and in photosynthesis, they do not release, that is, oxygen is not released. Is it? That's why these are known, this type of photosynthesis is called non-oxygenetic, that is photosynthesis. Is it right? Non-oxygenetic photosynthesis, important one. Come to the next now. Yes, chemosynthetic bacteria. These are also autotrophs, as I told you earlier. These are also, that is, autotrophs, but they obtain energy for synthesis of their food, that they oxidize certain substances. Inorganic substances are being oxidized. For example, this is ammonia, this is nitrate, nitrite, and ferrous ions will be oxidized. You see, during this oxidation, during this reaction, that is oxidation process, what will be done? During this oxidation, the energy is released. Is it? And this energy is used in formation of, that is, ATP. And this ATP are used in fixation of carbon dioxide, that is, food is formed. So energy is coming from chemical reaction. So do you agree that any sort of photopigment is required here? There is no need, because solar energy, light energy is not to be trapped. They do not utilize light energy and therefore there is no photopigment. Is it clear to you? There is no photopigment. They play a great role in recycling of nutrients. You see, nutrients are being blocked therein, that is organic compound. The dead organic compound, you see that this is dead organic compound. Dead organic compound, you see, that is the nutrients are being blocked. And these are the, that is, chemoautotrophs, that they will simplify it and that is minerals will be released. Right? You see here that some main types of chemoautotrophs are, this one is hydrogen bacteria, as the name indicate. These oxidize hydrogen into water. Example is hydrozinomonas, this is in italics, you know it. Nitrifying bacteria, these oxidized ammonia into nitrate. Two steps are there. First, ammonia will be oxidized to nitrite. The two important bacteria are, one is nitrosomonas and the other one is nitrococcus. And now, nitrite will be oxidized to nitrate. The two important bacteria are, one is nitrobacter, one is nitrocystis. Is it? You see, that is, this is nitrosomonas, this repetition is there. You see here, the third one is sulfur bacteria, but before that I'll ask one question. What's the role of nitrifying bacteria? That question usually confuse. Nitrifying bacteria, they do not fix, that is nitrogen No. Nitrogen fixation is not done by these, that they will convert ammonia into nitrite and then into nitrate. Is it? Sulfur bacteria, they oxidize sulfur or or hydrogen sulfide, oxidized sulfur, but will be formed either that is sulfuric acid is formed or that is hydrogen sulfide is formed in other process. These are 𝕆𝕆𝔻 oxidant, that is this is one name, and the other bacteria is that is 𝕆𝕍𝕆. Iron bacteria in the same way, they convert ferrous ions into ferric 1, oxidation is there. The important examples are ferrobacillus and that is leptothrix. So you see children, yes, a part of this chapter, that is chapter number 2, part 1, we have finished right now, in which we have discussed that is 2-kingdom classification, 3-kingdom classification, 4-kingdom classification, and then 5-kingdom classification in detail. That is a synopsis I had given to you for 6-kingdom classification, and then we have seen that is kingdom Monera, a part of this, and you have realized what that is. The Monera or bacteria, these are simple in structure, but these are very complicated in behavior. They have that is metabolic diversity. Thank you very much. In next lecture, we have to discuss that is second part of this chapter. By that time, have a nice day.